meiosis and genetic variation. This is for you grade 10. This is chapter 6, section 6. Last time we talked about meiosis and how this division only occurs within your sex cells. Okay, it leads to the production of gametes, and gametes are the sex cells, and it leads to the production of cells with half the number of chromosomes. And these cells with half the number of chromosomes are going to meet the other sex cells with half the number of chromosomes, leading to the formation of a full individual with full number of chromosomes. One of the main aims of meiosis is genetic diversity. What is genetic diversity? It means that the genes are mixed and matched during meiosis, leading to the formation of individuals that are not a copy-paste picture of their parents, whether these individuals are plants or animals or humans. So the main aim of sexual reproduction is having individuals which are not identical to their parents, although they carry their genes. The homologous chromosomes that pair up randomly along the cell equator during meiosis 1, it's a matter of chance which of the two chromosomes from any homologous pairs end up on a given side of the cell equator. So they line up on the cell equator and then they get separated at this point. Okay, so the allele pairs are segregated, means that they are separated during meiosis. For us humans, for example, a sperm cells with almost 8 million chromosomes combinations, when it fertilizes an egg cells which almost has also 8 million chromosome combination, we usually have an individual that is totally different from his parents, although he's carrying all his parents' genes. We have an uncountable number of combinations of chromosomes at the end. The independent assortment, which means that the genes are inherited independently, and the fertilizations, they play key roles in creating and maintaining genetic diversity in all sexually reproducing organisms. However, the number of possible chromosome combination varies by species. The probability, for example, that a bald eagle or a rabbit-eared bandicoot will inherit a specific allele is determined by the same way that it is for pea plant. Again, Mendel used the pea plants because they were easy. He could cross them easily. He could count the number easily, but he used them in order to get to these results. So we're still talking about how meiosis, this division, leads to the diversity in genes. We don't want, for example, a parent with a certain disease to give this disease to his offsprings. So this diversity will only happen in case of meiosis, and this diversity, it prevents the diseases, for example, from being permanently present in a certain family. So if a person from a family with a certain disease marries a person from a family which doesn't have this disease, the offsprings have better chances not having the disease. Another thing that leads to improving the genetic diversity during meiosis is the crossing over. The crossing over is the exchange of chromosome segments between homologous chromosomes during prophase 1 of meiosis 1. At this stage, each chromosome has been duplicated, the sister chromatides are still connected to each other, and the homologous chromosomes have paired with each other. When homologous chromosomes are in this position, some of the chromatides are very close to each other. Part of one chromatide from each chromosome breaks off and reattaches to another chromosome, as you can see here. The crossing over here, see, this is a crossing over. Part from this chromosome, it is going to detach and go to this one here also. So if you see, this is the blue one, and these parts are the parts that had a crossing over, and we call them recombinant chromosomes. The crossing over means that there is more and more mix and match of the genes in order to improve the genetic diversity. Because crossing over results in new combinations of genes, it's also called recombination. The term recombination generally refers to any mixing of parental alleles, including recombination events other than crossing over. Okay, so now you know that about crossing over. We're going to talk again about the Mendel's experiment. Mendel, all what he did was counting numbers, crossing the pea plants, and getting to know how different genes are inherited from the parent to the offsprings. So after learning about meiosis and after learning about crossing over, will the genes, when the two genes that are both on the same chromosome be inherited at the same time, or will they display independent assortment? In this case, the answer will be actually it depends. Okay, the farther apart two genes are located, the more likely they are to be separated when crossing over happens. So the genes that are close to each other on a chromosome, they are going to be inherited together. The genes that are away from each other on a certain chromosome, they are going to be inherited separately, and they might not be apparent at all in a certain generation. So the genes that are located close together and tend to be inherited together, this is called genetic linkage. So genetic linkage means that the genes that are close together on a certain chromosome are going to be inherited together when the meiosis happens. Linked gene will be inherited in the same ratios as a single gene because they are not going to be separated. On the other hand, the genes that are far apart are more likely to assort independently, which means they are going to be inherited independent of other genes. That's it with the genetic linkage and the meiosis, and this way we finished chapter 6, and this is all what you're going to have to study for your fourth quarter.